Hello everyone, so this is going to be the final experiment for our semester-long project, and it's going to be based on the kinetics assay. So last week we talked about the activity assay, and what we wanted to know is our beta-gal active? And assuming that it is, this week we want to determine how active our beta-gal is. And so we're going to do that using a kinetics assay. So our kinetics assays are based on inhibiting a protein kind of gradually, so adding more and more and more inhibitor and just plotting this data, and I guess in terms of reaction rate versus substrate concentration. And this kind of system is based on the Michaelis-Menten kinetics model. So here you can see I have the formula for the Michaelis-Menten kinetics. And I'm sure most of you have already seen this formula once before, but just to quickly talk about it again. We have V, which is our rate of reaction, equal to the V max times the S, which is our substrate concentration, divided by the Michaelis constant plus another substrate concentration. And what this Km is it's basically the amount of substrate needed in order to reach 50% of Vmax, so the 50% of the rate. So if we plot this data on a graph, this is what you should expect to see. So on the y-axis we have our reaction rate, on our x-axis the substrate concentration. So at low concentrations we have a very low rate of reaction, and as we increase the substrate concentration, the reaction rate will go up, 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 up until eventually it starts to plateau. And you can think that this plateau occurs because basically our proteins are flooded with high amounts of substrate. So just kind of imagine we have our enzyme, it takes in a substrate, forms into a product, and just as it kicks out that product, another substrate comes immediately into the enzyme. And that's what we should expect at these higher concentrations, which is why we kind of get this plateau, because now our proteins cannot work any faster than where it is right now. So for us to run our kinetics assay, there are really two types of inhibitors that we can use. We can use a competitive inhibitor or a non-competitive inhibitor. And so how they affect the graph, all you guys really need to look at is the Vmax as well as the Km, because each inhibitor will affect one of these primarily. So starting with the competitive inhibitors, as the name implies, these are the inhibitors that directly compete for the active site. So here you can see that we have our gray enzyme with a triangular active site that fits not only our substrate, but also an inhibitor. So because the substrates and the inhibitors will likely compete and try to take this active site for themselves, it's going to slow down the reaction. Meaning we'll need a higher amount of substrate in order to eventually dilute out this inhibitor in a way. So if we have 10, 100, 1,000 times the substrate than inhibitor, eventually this inhibitor becomes 0, and we should have the same Vmax. But if you think about it, that also means for us to achieve the, 1/2 Vmax, or yes, in order for us to achieve the half Vmax, we also still have to increase the substrate, which increases the Km. So for competitive inhibitors, you need to know that it primarily affects the Km of the reaction. For non-competitive inhibitors, are inhibitors able to bind somewhere else on the enzyme and inhibit it in a different way? So here we have our same enzyme with a triangular active site that can accept the substrate but not the inhibitor. However, this enzyme also has another site that fits our inhibitor perfectly. And if this inhibitor binds to this circular area, it may change the active site to the point that our substrate can no longer bind. So you can think that if this inhibitor binds and changes the enzyme, then this kind of completely removes the enzyme out of the entire reaction. So let's say we have enough enzyme to remove 50% of the enough inhibitor to remove 50% of the enzyme. Then we can expect for the rate of reaction to be kind of cut down by half. So this will change the Vmax. However, because the active sites of whatever enzymes that are available remain unchanged, their rate of reaction should still stay the same, or their Km should really stay the same. So it's a little confusing when I just talk about it. So here I have the effects kind of plotted onto a graph. So in green, we have our actual enzyme working with no inhibitors. You can see how it makes the proper slope and eventually reaches its Vmax. When we talk about our competitive inhibitor, we can see that the Km has actually changed because now we need more substrate to achieve this 1/2 Vmax. But as we increase the substrate concentration, eventually we dilute out our inhibitor and are able to reach that Vmax. Now, if we go to the non-competitive inhibitor, no matter how much substrate we, add, we'll never be able to achieve higher than this new Vmax. And this is because we kind of removed a good chunk of enzymes with these non-competitive inhibitors. So we're working at maybe 30% efficiency than what we were supposed to. However, what we should expect is for the Km to kind of stay the same. The halfway mark for our new, our new non-competitive model should still be the same as the Km to our uninhibited model. So how exactly are we going to utilize this information for our beta-gal kinetics assay? So here I have the actual reaction for beta-gal. We have lactose, we use beta-gal, and it breaks down to glucose and galactose. Instead, we're going to use a different substrate. So this is a substrate that looks very similar to lactose, enough that it can trick beta-gal and bind to its active site. So this formula, this chemical, if you guys don't remember it, is IPTG. So remember that we used this for our induction so that we could get, so that we could remove a repressor because beta-gal could not hydrolyze this compound. So we're going to be adding IPTG into our reaction, and we should be able to see that the rate will eventually change depending on how much IPTG we add to it. So that's kind of the idea of the kinetics assay. We really just want to see how these lines end up looking when we have it uninhibited versus when we have it inhibited. So if you guys have any questions about the actual experiment, let me know. We won't really be able to perform the experiment, so you guys will only have to do the quiz for this. And that'll be pretty much it for the rest of the semester. The last thing you guys need to do is just start working on your presentations. Remember, they are due, I believe, on December 1st or November 30th, sometime the end of November. So please try to get those in on time. And if you need any help or need any more information about anything about the lab, please feel free to email me.